All right, let us do a couple of quick things. Um, I'm just out. Fuck, my eye is itchy. Okay, so we're gonna just get the word out that we are doing some rearing rambles because who doesn't adore rearing rambles? Hang on. Here we go. Share. Copy that link. We're gonna say, all right. One minute. Okay, friends. All right, let us put this up on Lepidoptera. Let's see. Fuck, I did something wrong. 1 second, I'm an idiot. Okay. I already knew that. Lepidoptera. Post. All right. Where's the other Lepidoptera group? It's this one. All right. Word is out. So, let's talk about something. So I just realized something kind of important about myself, but my approach to humanity is a rearing approach. Yeah. And it's certainly a trauma response, and I'm working on it, but we're not going to get into that right now. We're going to get into lepidopterans and their host plants. Now, the first thing I'm going to get out of the way is the fact that Lepidoptera are not excluded to or confined simply to external folivores, okay? There are lepidopterans that are gall formers and leaf miners. I've reared lepidopterans from a few other feeding guilds. For example, wax moths, the Galeria melanella is an insect that eats anything you would find within a beehive. That includes beeswax, that includes pollen, that includes dead insect material, and all sorts of other good shit. Like honey is, a primary like carbohydrate source in the diet of Galeria melanella and several other pyralid moths. So not every lepidopteran species on the planet is going to feed on like a tree branch, like the leaf material. There are species that are decomposers. There are a few parasitic species. There are species that are aquatic and they skim and feed on like biofilm. There are species that mine and bore and do all sorts of other things. So they basically can be a number of different feeding guilds. And when I was teaching an entomology class, I had the students consider that caterpillars in a concealed feeding guild tend to not have a lot of pigmentation, because why the fuck would you bother having pigmentation, which generally only does you external pigmentation, like that you store in your skin cells, doesn't do you any good without sunlight. So if you're not being bombarded by UV rays, it's a little bit pointless to have an excessive amount of pigmentation. Furthermore, if you're in a concealed feeding guild, there's less of a selection pressure to visually hide from predators, because of the fact that you're just not out in the open in the first place. Luna moths, for example, have this incredible plasticity in the appearance of laden star larvae, based on what they're eating. And they more or less match the pattern of vein on the back side of their host leaf. So if you're feeding on walnut, you're going to see this big, mostly green caterpillar. You're going to have very few of the bumpy scoli that are different colors, like you might see on a larva that's feeding on hickory or black gum. Now, hickory and black gum are prone to a lot more galls in this region, and the edges of those leaves, especially in hickory, tend to take on funny splotches of brownish coloration with age that walnut just doesn't. And so when the Luna moth larva is feeding on hickory, the scoli on the back and on the sides have a lot of these orange and pink colorations. It's really quite amazing. And larvae that are feeding the fall have a lot of brown and yellow coloration to them to kind of match the background of what might be going on within the host system, because a plant doesn't turn from green to everything is fall color overnight. You're going to have pieces of leaf matter that are still green. You're going to have some branches where the leaves are still green, whereas other branches of leaves have fallen off. Sweet gum does this to quite an extent, where you can actually collect green sweet gum leaves in November in Rhode Island, especially if we've had a very mild fall. A lot of the plants in New England are kind of adapted to pick and choose when exactly they start and end their seasons. And that's because some springs are really, really warm, and you have thawing, in like the second week of March, and then a week later the wood frogs breed and everything. Or you have like what's going on this year, where like a lot of things are late. A couple ferns spoiled a little earlier than expected, and Cypripedium macaulay was a little later than expected. It's just been a weird year for a lot of different things. Lunas were late this year, and that almost never happens. If lunas are late, something is really wrong. You know, um, one of the things is that I have an appreciation for a species, regardless of its abundance or rarity, because an organism may not necessarily be abundant or rare, um, simply because it's a specialist or a generalist. There could be a number of factors. And it gets me kind of wondering, like with something like Actius luna, where like I'm sure you have these subpopulations throughout its native range that kind of do one thing, and you know, every species kind of behaves differently across its native range, and we'll come back to it when we talk more in depth about lunas specifically. But even though luna moths are frankly a dime a dozen, you don't have to put a lot of effort into obtaining Actias luna. Like everybody has them if you can't collect them from the wild, or females will come to light to a degree that you don't see in very many other Saturnaid species. I suspect that lunas travel quite a distance on their nightly flights, and then a light might intercept them. Because even though when you zoom out of New England, it's more or less a contiguous forest, there are definitely stretches where suitable habitat for lunas to really do anything are quite far apart. And to the degree where a luna moth would reasonably be crossing several neighborhoods to get to another habitat patch, and that's where they come to a light. But meanwhile, there are species where unless you have like a patch of the host plant 2 or 3 feet away when you set up your light, you're not going to ever see a female. There are species that are like that, okay? And then there are things that are kind of like in between where they just kind of do their own thing. Anyways, so when you rear folivorous lepidopterans, one of the things about the lepidopterans that feed on leaves is that they are not as reliant on the host material being alive, because they're eating tissue that more or less has an outer dead layer and a little bit of metabolic activity in the middle that is really just redirected elsewhere in the plant, because plants kind of put out leaves with the intention of them wearing down and dying over time anyway. So to the plant, losing some leaves is the least of its concern. And as a result, now, caveat here, I'm not saying that host plant health is insignificant in the development of folivores, okay? Of course it is. What I'm saying is that should you attempt to replicate what will be happening in nature, the amount of diligence required to keep up with making sure that the insect has constant access to fresh food material is most easily achieved with folivores. And the reason being, folivores, especially external folivores that are just adapted to be relatively mobile, okay? They don't really some may make a small silk web to hang off of, but they're not like inserting half of their mouth into like a leaf vein or a stem or anything where if they're knocked the wrong way, you may damage them. They kind of just, stick on the outside of the host plant, and they kind of just eat what's jutting off of the host plant that's just there. And they can just move around eating their home. And so things that form galls, for example, unless that gall former is, going to complete its life cycle in just a few days, the host plant needs to remain completely alive and relatively intact. Same goes for phloem feeders, to the extent where you may have to cull large portions of less motile aphids. Aphids are kind of hard to culture because if you let them expire the host plant, your colony can crash. But you do cause problems by disturbing the colony quite a bit. And so what we would do with our lily aphids was we would, have them colonize the lily. These lily aphids, if sufficiently disturbed, and that could mean, you shake the crap out of the plant, or maybe they smell the better, fresher plant material nearby, and then they're like, "Hey guys, let's go over here." They just, things like lily aphids don't like set up shop exactly on the, like, they're able to move away onto new plant material. A lot of aphids can, but some aphids can't. So usually the aphids where you can have them colonize a plant, and then when the plant starts to look a little bit yellowy, you cut the plant up, and then you put each piece that has, maybe a few dozen aphids on it onto a fresh new plant. And, you just keep that cycle going, culling off whatever excess, and, we would be rearing the aphids mostly as feeders for mantises and ladybugs and all sorts of other things. So it wasn't like we were just saying, "Oh my God, I really want to have an aphid colony." No, we were using them more or less for all sorts of things. Hatchling assassin bugs will eat aphids quite well. And you do want to have healthy prey items, and that starts with healthy hosts. But my point is that when I rear lepidopterans that are external folivores, and that is a great many lepidopterans that people pay attention to. Most of the lepidopterans that exhibit or inhabit those other feeding guilds, such as galling and being a leaf miner, these are like microleps that are like, as adults, the size of a fruit fly. They're that tiny, where only a couple of people who really like, a lepidopteran that you need like a jeweler's loupe to appreciate. They are really cool. I really want to try and rear one day, but in any case, it is possible to rear leaf miners, as I have learned. But when I rear external folivores, I oftentimes will need to figure out most ideal hosts depending on what kind of rearing I'm doing. And what I mean by that is, some leaves respond better to being cut off of the plant than others. Some leaves, like mulberry and sweet gum and oak, if you cut them off the plant and, clean them off and make sure that they're then, wiped dry, you can put them in a freezer bag within the crisper drawer of the fridge for about 6 weeks, and they're still nutritious enough to feed your larvae. And then other things like sassafras wilts within a few minutes to the point where something's not going to want to eat it. So just because the species I'm working with can eat a plant doesn't mean I'm going to use that plant for indoor rearing. And one of the things that you have to understand is that sometimes you don't have a choice but to rear indoors, especially if it's, a species like anything that eats tulip tree. You can't really sleeve a tulip tree. These things are, 80 feet high or taller, and you're gonna have to go, the lowest branch is 20 feet off the ground, and maybe there's a seedling around, but no, the only seedlings aren't on my property or something like that. And because they're not on my property, they cannot be sleeved by me without repercussion. Things like that are why certain host plants are not practical for sleeving. Or, if it is a species that's not native, you shouldn't be rearing it outdoors. I don't think I need to stress or emphasize or elaborate on that. You really don't want to have any non-native species just roaming free, especially not animal species. I mean, to a degree, not plant species either, unless, it's one of those plant species that so sweet gum is an example where an established mature tree of sweet gum can survive quite a bad winter, to the point where you'll find them planted all throughout New England, sometimes into Maine, and they survive the winter. But it's too cold for young, first year seedlings to survive the winter. And that's why they're, for now, an okay tree for some of that, because they're not going to, quote unquote, escape cultivation. But it's a lot harder to stop an animal, especially a moth. Okay, remember how Lemantria dispar started with a couple of loose adults that some guy. The reason we have Lemantria dispar is because we didn't understand genetics all that well back then. And in all fairness, Lemantria dispar and Bombyx mora used to be in the same genus. So the guy had some rudimentary understanding of biology and how hybridization worked. However, our knowledge of the taxonomic relationships between things like the Erebidae and the Bombycidae, with the Erebidae being more closely allied with your noctuids, those army worms and cutworms and those guys, and then your Bombycidae being closely allied with the giant silk moths and things like that. They're nothing alike, but because of how limited, in all honesty, insect taxonomy was really bad in Linnaeus's day, partially because of a bias against those types of animals. Now, I don't want to dive too much into that, but I suspect that the reason why there was no oversight into, why a whole bunch of things were arbitrarily placed in the genus Bombyx, and then a few other things were arbitrarily placed in the genus Sphinx and whatnot, was partially because there just weren't a lot of entomologists who had really come out of the woodwork in Linnaeus's day. And so then several centuries later, when whatever this person's, I don't know the guy's name, but he brought Lemantria dispar over from France because he wanted to, create a silkworm that would produce a good cocoon, but that would eat more than just mulberry. That was basically the intention. Now, this is where sort of the Dunning-Kruger effect comes into my mind, because the person had the, sort of knowledge of some of the, aspects of how hybridization may or may not work, maybe not in inheritance, because we didn't really understand inheritance patterns then. Basically, you can't breed two organisms together just because you want to have the desirable traits of both of them to be represented in their offspring. That's just not how that works. And the reason why is because the traits that are most beneficial to them aren't necessarily the traits that are most convenient to us. And until we get that through our thick skulls, of course, we're going to be doing these ridiculous willy-nilly things like trying to induce hybrid vigor in things that were ultimately too far apart to even be capable of mating if they wanted to. Okay? So somehow, the Lamantria got out of his apartment or whatever, and he was unable to contain, the subsequent larvae, and they started an infestation, and they've just been eating away the East Coast since. Okay? And so you really have to be especially careful with non-native lepidopterans, particularly ones that are native to temperate regions. I'm less concerned about like Atlas moths and some of the tropical samia getting out in Rhode Island and surviving a Rhode Island winter as I would be concerned about like near the what's it called? Daphnis nerii or something like that. I mean, Daphnis nerii is generally confined to the Mediterranean during the winters. However, there are United States native Sphingidae, such as Eumorpha acmon, that are confined to the southern states during the winter months and that are occurring in mid to late summer up here after they migrate further north in subsequent generations. That's what's happening, okay? And because you have species in Europe that have a similar way of functioning, you have to be kind of cautious. As interesting as some of those European sphinges are, we have some just as interesting, native North American ones. Now, that aside, the other thing is that, sometimes you're rearing a specialist where the host plant that it feeds on just isn't occurring on your property, or something like that. So, sleeve rearing would be impractical, or it's a species where, it lives on like a rosette forming host. And, it is a pain in the ass to try and rear anything on something that's just a fucking rosette on the forest floor or something like that. It can be done. And, I strongly recommend like you take the like rosette and you like dunk it in like a bucket of water and you wash it first so that you dislodge, you want, kind of to not have too many dirt particles in a rearing container indoors, okay? Lepidopterans don't, well, at least the foliage feeding lepidopteran species don't rely on a gut microbe in order to go ahead and process food anyway. They just don't retain a resident gut microbe. So, you can rear lepidopterans under more or less aseptic conditions, certain species, without causing too many problems for them, at least from a digestion perspective. Now, that being said, I'm not advocating for entire sterility. I'm advocating more for cleanliness that keeps in mind that foreign particles should be kind of vetted to prevent, pathogens, because these animals are living under artificial conditions that are stressing their immune systems anyhow. Things that wouldn't be pathogenic outdoors to a caterpillar are pathogenic indoors to a caterpillar. But you also have to recognize things that are not pathogenic to wild caterpillars can become pathogenic to caterpillars that are too crowded. Okay. Some species, especially like Hyalophora cecropia, for anybody looking to rear Hyalophora cecropia, you need to keep them under very low densities. I mean, the first couple of days, fine. Like I just had a big hash out in a cup, and like right now they're all still together, but they really haven't even started feeding yet. So they'll be fine until tomorrow. They'll be just fine. And then tomorrow, what I'll probably do is, they have a couple of cherry sprigs. I'll pick off each leaf and put it in its own cup and give each cup new cherry. And so that'll be one way of just kind of helping to minimize the density. And, that seems to work pretty well. And then I'm going to have to get into, all right, we're going to have to leave them or, something of that sort. High Leffer supposedly can be reared relatively successfully individually indoors. I've done it before, so that is another option. I'm anticipating, maybe somewhat higher losses, but it's still doable. Some species, it really is just a matter of not being crowded, not being near one another. But anyway, so if I rear species that are like tulip tree specialists. I'm rearing a tulip tree specialist right now, and we'll get into that later. But if I'm rearing a tulip tree specialist, I have to rear it indoors. So I need some way of verifying if all of my tulip tree specialists die indoors. Is it because I did something wrong, or is it because there's just something weird about tulip tree when you cut it and you put it in a container inside that makes it unsuitable to rear on? Because that is a very possible thing. Now, what I'll say is that because I have no issue rearing Papilio glaucus, the eastern tiger swallowtail, on cut tulip tree indoors, I would venture that if Calissaemia anglifera, the tulip tree specialist, languishes indoors, it's probably not a result of the tulip tree. We also have had Illinois Illyriodendrini do just fine, at least short term, on cut tulip tree, and that's an aphid, by the way. But anyway, so some plants are temperamental when you remove them from, the whole system. Things like beech, things like ailanthus. You cut it and, it just like really just, it doesn't wilt because it's still got a lot of water and it's tough foliage, but you put it in a container and then after a few hours, it's just nothing but condensation. When you rear lepidopterans indoors, you generally use non-ventilated containers. And the reason why is because a lot of these animals are adapted to living under relatively high humidity. Plus, you want to make sure that the, host plant material that you've cut doesn't wilt. And it's generally not safe to maintain Lepidoptera larvae in a container with like standing water to, maintain host rigidity. So the best thing to do is to have slightly moist host foliage put in a container and there's sufficient airspace, and maybe the container, even though it's closed, isn't 100% perfectly airtight, as most containers actually aren't anyway. And that airspace is sufficient to allow gaseous exchange and whatever else that has to happen. And, with you regularly, once or twice a day burping the container where you open it and allow just the air to kind of rotate, that's plenty for enough, you're not getting them suffocating, for example. Their metabolic rate isn't as rapid as, say, a mammal. If you put a mammal in a container like that, it's probably going to suffocate. So please do not do that, okay? I do not advocate for suffocating mammals. Do not put a mammal in a fucking sterilite bin. But you can put a caterpillar in a sterilite bin. You can put a small enough frog in a sterilite bin, okay? Once the frog gets to a certain size, don't do it unless it's for transport. But, a sufficiently small frog would be just fine. Now, anyway, um, don't so the metabolic rate of these animals isn't so rapid. Um, the caveat there is, because there is, also the metabolic activity of the cut foliage, there still is that CO2. So you can't leave it closed indefinitely, but you can definitely leave it closed for 12 to 24 hours without worrying about the animals and they're suffocating. If they suffocate in there, they're probably too crowded to begin with. And, you don't want to crowd things to the point where they can't sit together in a container without using all the oxygen. They need to. That is just too crowded, not just because of the carbon dioxide. It's also too crowded because of things like waste accumulation and competition for food, because a handful of cut host leaves or whatever is going to be used up by 5 caterpillars much more slowly than 400. Okay, especially if they're all in the same instar or the same size. Maybe 400 hatchlings versus 5 fifth instar. That's a different matter. But 400 fifth instar are going to use up a whole lot of host material. So you don't want to have them all in the same container anyway. I may have to take a quick break to refill the water. But anyway, so at any rate, you need to kind of get a feel for some of the best ways to maintain insects on hosts. Now, one workaround that is oftentimes utilized is to have, for species that feed on relatively small host plants, the caterpillars just maintained on a potted host. Now, aside from the potential hazard of the potting soil to the larvae, which is easily worked around, that's not a terrible way to rear the larvae. One caveat I have there is that when you cut host foliage, you don't give the plant a chance to kind of respond to the fact that there are caterpillars feeding on it, whereas when it's on an intact rooted plant, you do. And under more normal circumstances, less stressful circumstances, that might be just fine. However, you may have reduced rearing success on potted plants indoors, especially if they're growing, in fertilizer-rich, very well illuminated, frequently watered settings. Although, folivores are more prone to the plant vigor hypothesis inherently, but that's kind of aside from the point right now. Sometimes you can do fairly well actually with potted hosts, and that is actually a good way of answering questions about host plant response to herbivory in ways that you can't necessarily unless you're sleeving larvae on the intact host outdoors. But then when you're doing that, it's a little bit harder to control for other environmental variables. Now, remember that the good is the enemy of the perfect, so that might not necessarily be the worst thing to worry about. But nonetheless, if it's not practical to rear the larvae on a potted host indoors for one reason or another, and that could be something like, you live in an apartment where you can't necessarily maintain, a lot of large cages with bright lights over them to keep the live host happy and the larva happy, because sometimes the more trophic levels you add to your setup, the you know more ways you can fail and everything collapses. Now, I've learned this, the hard way many times. It's something that you roll with. But what I'm getting at is there are plenty of reasons why it may not be practical to have a potted host that is, within a larger cage that accommodates the host plant and the larva. Because the last thing you want to do is make the host plant totally die, in the process of feeding it to your larva, especially if that's the only thing you're relying on to feed your larva. With that being said, there's this sort of other option where you somehow cage or so like you cage or sleeve hosts out in their native area. And there are certainly artificial diets people employ instead of using host plant material. But let us just continue to consider that I need to know if, a commonly available host that, hey, maybe I can use in a pinch, or if this is, if I'm trying to reach something that specializes on it, I want to see if a generalist will do fine indoors on it. Now, I'm aware that it's not a perfect comparison because things will utilize that host foliage in different ways. And so cutting that host foliage may affect something that's a generalist feeder less than it would affect a specialist feeder. So it's not perfect. I agree. But just in terms of base level, how does that host respond? When you cut it off the branch, is it wilting before I can even get it to the larva? Or, does it exude so much water that it drowns everything in a container or something like that? Or, there are other things. Sometimes it loses nutritional quality without necessarily without changing visually, which can be really confusing. Oak and hickory do this quite a bit. And so I try to be careful rearing things indoors on hickory and oak, especially hickory. I really just don't bother rearing anything indoors on hickory because if it'll eat hickory, it'll eat walnut, and most things do just fine indoors on cut walnut. As long as you're diligent enough about keeping the container clean, walnut does lose water quite rapidly and does kind of sweat a little. Not to a degree that can't be managed, just by being a little bit discretionary about how much cut foliage material per volume of airspace your container has. So you know what? I have an idea. I'm going to take a quick break to refill my water bottle, and then I'll come back and we will talk about something interesting. I'm back. Okay. I have my water refilled. Now, we were talking about host plants and practicality of having caterpillars maintained on host material that's been like potted and is growing under artificial conditions like that. And so we were talking about walnuts, okay, and how like so here's what I was gonna get at before the break. Why don't we just think if you were to take 2 cotton balls, right? You'll need 2 cotton balls. You'll need, I don't know, a 2-ounce deli cup, and you'll need a 32-ounce deli cup. Both of them should, the 2-ounce portion cup and the 32-ounce deli cup should have airtight lids. What I want you to do is put a teaspoon of water on each cotton ball and put 1 cotton ball in the portion cup and put the lid on it, and put 1 cotton ball in the deli cup and put the lid on it. Then put it on a sunny windowsill for about 36 hours. And then what I want you to do is I want you to open up each cup and kind of just give each cotton ball a feel. And what you should notice is that the one that was in the 32-ounce deli cup should feel a little bit less saturated with water than the one in the 2-ounce portion cup. And that is simply because there is more airspace for the water vapor to kind of dissipate into, which means there's going to be more evaporation, and there's going to be more, sort of air exchange. So when you're rearing things on walnut, the way you kind of deal with, walnut's kind of tendency to perspire is you just kind of rear things at a little bit less of a, lower density, a little bit bigger container, and you'll be just fine. Things like beech and ailanthus, they really don't give you even that grace, like walnut will, if you put a whole clump of walnut into the cup, you're gonna get a lot of condensation, and it's gonna be really okay. That was getting distracting, unfortunately. Anyway, I just had to now what you'll know is that the dissipation of moisture from a surface is going to more or less be constant. Now, all right. Sorry, I just need to catch my breath. Now, what you end up with is the need for maybe trying to see if a host behaves relatively well. So, what I generally do is I try to figure out what sorts of proxy species might be helpful. Like Actaeus luna is something that is pretty darn good for a lot of host testing just because it does very well indoors. And so if you have, Actius luna, you're rearing indoors, and so to the live chat, I host test when I'm host testing, I am just kind of testing the suitability of a host for indoor rearing of a moth. And so what you basically end up doing is you have this is what we're getting into is you'll say, I want to rear a specialist that will only feed on whatever host. And because I can't, rear it outdoors on that host, I have to rely on, the cut foliage from that host inside. And so I might, before trying to rear that specialist on that host inside, rear something that I know does well inside, a generalist that will eat that particular host and do just fine on it indoors, just to kind of, see about where we're at with, okay, this is not a temperamental thing. And again, as I said before, it's not perfect because some organisms, some species may be better adapted to, less nutritious leaf material than others, or maybe leaf material changes in that short time from being cut to, being replaced, that it's just less suitable. I just don't really know. And, it doesn't you don't necessarily have to know all of that just to kind of get a general idea of how easy it might be, how user friendly that host plant might be. It doesn't necessarily mean that it's nutritionally superior. It just means that, when you cut it and you put it in a container, it doesn't end up like condensating all over the place and killing your larvae or doing some other stupid thing like that, whatever that stupid thing might be. And so right now I have Actius luna, the luna moth, eating black gum, which is Nyssa sylvatica. And Nyssa sylvatica is one of those interesting native trees. You really, you'll only really find it like in really wet areas, kind of like woodlands that are adjacent to like a wetland where, the water table is pretty high, and so, the roots are, more or less inundated, even if the ground never floods at our level. So, black gum has this kind of really like dull, kind of khaki grayish bark, and it's generally maybe, a foot in diameter or less. But the defining feature is its branches are comically thin, maybe like the thickness of your pinky, and they jut out of the trunk at random at 90-degree angles. It's just the most ridiculous, hilarious thing. It's a very distinctive tree. Now, I'm sure there are a lot of other things that'll feed on it. Various geometrids come to mind, and it might be a good alternative to oak and sweet gum. I don't know just yet, but oak can be a little temperamental with things that feed on it indoors. Sometimes I don't use oak for things indoors just because a lot of oak feeders that'll eat other non-oak things, they just stress out when they're reared on oak, and that makes them more susceptible to disease. And I don't want things just getting diseases. And so what I do is I rear a species that I know how to deal with indoors in rearing bins. And I have quite a few of those. Now, unfortunately, a few of them are specialists, but and then a few of them are forb feeders that don't necessarily um, help with what I generally work with. I mean, they're still interesting. They're just not necessarily the most useful in that context. Now, I will oftentimes rear things indoors for reasons such as better control over their environment, and less worry about predation, things like that. And if I don't have access to rearing sleeves or something of that nature, then I also then just have to rear them indoors. And so indoor rearing does have a lot of advantages, but there are a lot of things that just can't tolerate being reared indoors. What's that? Anyway. Now, you end up with this kind of curiosity about what sorts of things you might go ahead and rear indoors and what hosts you might go ahead and use. Yeah, I am in a lab in a college right now. So yeah, we're live from a lab. Yeah, I will. I think someone else is in the building. We'll be fine. I'm not too worried about it. Anyway, so I will rear something that I know how to deal with indoors that doesn't have a problem being indoors, on a number of different hosts to see what sorts of things happen. I want to kind of look at like what kinds of diseases show up and what proportion of larvae get sick and how quickly do they develop, and a number of other things. Are they reasonably sized when they're mature, and things like that, where I know what hosts you can rear like Luna moths indoors on. Like Sweetgum and Walnut are generally what my go-tos for my population in Rhode Island, where that's yeah, more or less nutritional ecologists mostly. Yeah, we do a lot. I do a lot of nutritional ecology right now, and so when you rear Lepidopterans indoors on cut hosts, and again, there are a number of good reasons to rear them indoors on cut hosts. You tend to require, a more thorough understanding of how the host plant you decide to utilize may affect their development. And so this is important to know because if I want to rear a specialist indoors, before I rear a specialist indoors, I want to kind of know what happens to a Lepidopteran when I cut the host material, when I take a pair of plant pruners and remove that branch from its source of nutrients and water. And I have the understanding that yes, in about 48 hours that material will no longer be acceptable to that larva, but it's not practical for me to be there's only a certain frequency at which going out and cutting fresh host material is practical. And if that practicality is like every 25 minutes, then you really are going to have a serious issue trying to rear it indoors. Just for logistics reasons, you might have the dedication, but just logistically it will be a nightmare. Now, anyway, I do want to, emphasize the importance of having an understanding of what's going to happen to that host material should you cut it off of the branch. And so using certain things like Samia cynthia is a saturniid that does quite well indoors and feeds on a lot of things that lunas won't. Cynthia will eat cherry and it will eat willow and it will eat a number of other things. Luna will eat some willows, but it's really hit or miss with Luna on willows. I just don't even bother because the things that they feed on, like walnut and hickory, are just abundant enough. But sometimes I will also want a host test done for other people's purposes. And so I am currently testing Actaeus luna on Nyssa sylvatica, not necessarily because I, for myself, plan to rear lunas and nothing but Nyssa sylvatica ever again, but the more host plants we can tell people are really suitable for indoor rearing of stuff. Even the most common, easygoing species like Actaeus luna, the better we're all going to be. So now we're going to go into a few specifics. So one of the things that you learn to do is you learn to understand that your moths are only as healthy as their host material is. And I go ahead and I have things I'm rearing indoors in containers on cut foliage, with the understanding that this is not the most ideal scenario for those insects. And I take note of things like, what's the mortality rate like? Why are they dying? Lunas have a couple of different diseases that they're affected by as they develop. Like the viruses don't tend to kill them until like the last couple of, weeks of development. But there's some disease that like makes the caterpillars just like get like this, they look sweaty and they die, but like they eventually get, this only happens before they hit a certain size, and then once they hit that size, none of them die of it. It's just the lunas have these weird things, and I take note of these as I rear things indoors, because maybe there are hosts that you don't see some of that weird shit. Now, lunas are unique in that despite, their love of dying of a disease of some sort for no good reason, as long as you keep things relatively like sanitary and you feed them fresh foliage, you're gonna get enough through to get another generation, especially if you're rearing more than like 50 caterpillars at a time. And due to that, you're not necessarily too panicked if you're rearing lunas indoors. The thing you have to remember is that most diseases you see in captive Lepidoptera larvae are confined to indoor rearing setups. Not all, and there are definitely health problems that can arise from outdoor rearing. For example, you can have all sorts of vertebrate animals manage to tear into your sleeves and remove and eat your caterpillars or cocoons. The other thing that can end up happening is the caterpillars can still something can go wrong inside the rearing sleeve very quickly, particularly overheating, especially with like Hylophorus cecropia. You can't really win with Hylophorus cecropia. You try to rear them indoors and they all die of a disease. You try to rear them outdoors, and the minute the sun hits the sleeve, they all die of heat exhaustion or whatever. Like heat waves kill them. I've had heat waves kill Cecropia larvae, even though they were sleeved in the middle of the woods. Cecropias are very temperamental. I don't know what else to say about them. Okay. Now, what I will say is that when I rear the New England population of Cecropias, they're a little bit more easygoing. But we're going to get into that later. That's a different episode. Okay. This is specifically about like what sorts of hosts do really well for things that do otherwise pretty well indoors when you remove them from the tree or whatever. Remember, I generally rear external folivores that feed on woody plants. Not everything that I rear is a feeder on woody plants, but the vast majority of stuff, yes, it is. So, what you have to understand is that is going to drive some of my knowledge, but it doesn't necessarily drive my approach. I would approach other feeding guilds similarly. Okay? I would. It's just that my current understanding, or the fact that I've caught on to the fact that, yeah, when you like some plant species just aren't the best when you cut them and you put something indoors in a bin on it. I'm not the first person who's taken note of that by any means, but I definitely have not seen anybody else do a deep dive on it. I would like to see that change, by the way. One of the things that you have to understand is that I don't necessarily view uniqueness as virtuous or good, because sometimes if I am unique in a way that might be beneficial to everybody, I would like to share it and thus no longer be unique in that regard. So when it comes to sort of, hey guys, I've taken note of this thing and, I'd like you all to know it, like, on one hand, yeah, that's kind of like I'm the first person to know that this thing happened, but that's not important to me. What's important to me is I'm not the last person to know that this thing happened. So when I catch on to something like, there are some plants that you just don't have good success when you like cut it off and put the material in a bin with a caterpillar. That's helpful to a lot of people. Now, what I can tell you is that there are a good number of host plants that, woody hosts that do quite well when you cut them. Grape and Virginia creeper are generally pretty good. Most things that I've reared stuff on isn't overly temperamental. There are things that are sort of like mildly temperamental, where like I'll still rear things indoors on it, because it's not so obnoxiously like ridiculous. But then there are things that are just exceptionally easygoing, like sweet gum and cherry, and things like mulberry. What was the other one? Yeah, things like that. And then things like yeah, grape is also pretty, reliable. You can fridge a lot of host material when you cut it. And then obviously some host material isn't going to keep for 6 weeks, but a lot of host material at a pinch can be refrigerated. And that is something that like I don't want to keep to myself. If I learn something in this, I don't want to hoard it. So I will share these observations, and some of these kind of drive me to want to do bigger, better stuff with it. I have to sort of remind myself that not everything has to be like some you know what 20x replicated peer reviewed study in order to make it useful. And I do tend to agree with that, because a lot of the when we do a lot of scientific work, and it's a more or less appropriate approach. But when you're doing science where you're like rearing caterpillars, you're generally you know not researching to improve caterpillar rearing methods. You're researching because you want to know if you know the sound of a wasp makes the caterpillars develop slower or whatever, something like that. That's why your caterpillars indoors generally. And so a lot of times the most efficient rearing methods are not you know necessarily um, what's it called, verified by scientific study. um, you know but it doesn't always have to be. That is the method, not the result, where you're like, say, that's just one of the methods. And as long as everything in the experiment is being reared under the same conditions, it doesn't actually matter. So no, there's really no need to have the most evidence-based. Now, it should be evidence-based, but you don't necessarily need peer review for a lot of rearing methods. Now, I will say there's always nuance. What you have to understand is that the efficiency of a rearing method is a very, very objective metric. People are free to have their opinions about my rearing methods of whatever, based on how they feel about the aesthetic value. That is valid and fine. But what you have to understand is that the evidence points to the methods I employ being, at the very least, efficient and 100% effective at assuring and maintaining the well-being of the organism, as evidenced by the fact that they are indeed performing well on it, well under those circumstances. Um, I had an argument with somebody at one point about whether or not I was feeding the crested geckos enough. And what I had to point out to them was that, I've been breeding crested geckos for nearly 10 years, and I've had no issues with animals, being unable to reproduce or anything like that, that were necessarily attributable to their diet. And so, the fact that these animals were capable of developing at a normal pace, reproducing in a normal, at a reasonable level, comparable and, quite frankly, identical to everyone else's, is an indication that, yeah, my methods are working just fine for them. But in any case, we are getting towards the end. What we're going to do, just kind of for the last few minutes before we wrap up, is talk a little bit more just about sort of how we got here and what I want people to kind of take away. So, generally, I will caution people when something seems to work pretty poorly for me. So I will rear things like Actias luna on all of the different host plant species they've been recorded to feed on. Now, I know that Actias luna will develop just fine well indoors. So if they do poorly on something I've never reared them on, it's not because they're being reared indoors, it's because something about that plant means that when you cut that plant and put it in the container indoors, it, kills your larvae. Okay, fine. Now I know this because if, for example, I want to rear something that I can only rear on indoors on this plant, I might need to refine my rearing methods or even just abandon ship altogether, depending on how crucial it is for me to rear that species. So, what I want people to take away from that is I find that beech and ailanthus are not the best hosts for indoor rearing. A lot of rosette forming forbs are also not ideal, just because as you're grabbing them, you're just pulling up a whole lot of other stuff. Remember that when you're rearing Lepidopterans on cut foliage, you don't want a lot of excess debris because you're getting a lot of things that are freshly dead for bacteria to start feeding on. You're stressing out your larvae already. You want things to be relatively clean. I don't mean absolutely sterile, but I always, do a 2% bleach mist of my host foliage before I give it to the larvae. And, this allows for just kind of the destruction of any viral particles and things like that may otherwise harm the larvae. And plus, chlorine is nutritious plant tissue, and so that may actually prolong the nutritional content of the leaf material for the Lepidoptera larvae. However, don't quote that as scientific fact. That is kind of just me thinking to myself and asking a question about whether or not that could possibly be the case. I'm not saying it is, but it is maybe possible. I don't know. For whatever reason, there are reasons why, a bleach dunk or mist or wash, helps indoor reared larvae. It's weird because you can rear larvae out on sleeved host materials where you put a mesh bag over a branch and put larvae on that, and you don't have any requirement to clean that host foliage with anything, and they do just fine. It is very possible that because the larvae indoors are under a greater amount of stress, they're more susceptible to infection, which is why you want to spray the host matter with the bleach. But again, things are not mutually exclusive. There are other chemicals such as ethanol you could spray on the plant tissue, but ethanol is more damaging to plant tissue. Chlorine bleach does not damage leaf tissue to the degree that you might expect. So a 2% bleach solution, it's going to dissipate off of that leaf before it has a chance to eat away at. Obviously, if you take a 2% bleach solution and you throw a leaf in there and you close the container and you wait a couple of weeks, of course it's going to eat away at the leaf. But what I'm talking about is you have this, branch where you're spritzing it with the 2% bleach, and then you're leaving it out in the air for just a little while, just so that the puddling is kind of dissipated away, but it's not completely dried off yet. You give it a good shake and then you put it in your larval bins, and that seems to work pretty well. And so forbs, things where you're plucking shit off the ground, just aren't the best for that end, if you will. Now, I don't know yet about black gum, but if anybody has any questions about whether a houseplant works well when you cut it and you rear something indoors on it, you are perfectly welcome to like comment or whatever, and I'll let you know like if I have any experience with it. I mean, I've tried a lot of things, but I'm sure there are things that somebody's going to say, have you tried rearing something on like hawthorn indoors? No, I haven't. Have you tried rearing whatever? There are things that I haven't tried when using as cut foliage indoors yet. um and that's to be expected. I'm not even, I'm almost 28, but I'm not 28 yet. I haven't been on the earth for so long that I could possibly have been like, um, okay. So I'm just kind of hanging out in yeah, I'm working in a lab, but this is a lab I'm just kind of, I have permission to be in. Like, I'll just come in here sometimes and watch Housebroken or something. But anyway, yeah, I'm open to answering anybody's questions at any time. If I've reared anything indoors on whatever, um, you know, sassafras and spicebush, I will say, are pretty poor hosts for indoor rearing on, just because they tend to wilt very rapidly when cut, and they lose their nutritional value so quickly. So I don't generally rear things indoors on spicebush or sassafras. Now, what I will say is that everything I rear outdoors starts out indoors generally, mostly because of earwigs. But like if I'm going to sleeve like Promethea larvae out on sassafras or spicebush, they're going to have at least a couple of days where they're eating the spicebush and sassafras indoors. And what I find is that the very early install larvae are a lot less picky about just how much water content is in that leaf tissue, which is why they're okay on cut spice bush and sassafras material for the first couple of days of, first week of life. But after that, you really do want to like actually get those larvae, put outdoors on the live host so that they do indeed survive better. Um, and so the takeaway here is to always remember that plants differ in ways that we may not necessarily understand. And so just because a caterpillar will eat an intact wild growing whatever host out in its native range doesn't necessarily mean that when you rear that caterpillar in an artificial setup with any degree of deviation, you're going to get the same result. Now, in general, it's going to be just a matter of have you cut the host material or not. And if you haven't cut it, then it's usually fine for most things under most circumstances. But what I will say, and I want to leave you guys with, is just kind of understand that a lot of these things are still full of unknowns, and they make us tick and they make us, a little crazy, but it's always fun to embrace it. And I encourage everybody to kind of, maybe ask a question and try to figure it out for themselves. And, you know, maybe do an experiment or two, do something fun. We're all kind of, on this planet one time, and we should all just kind of get to watch caterpillars develop or something. We should all rear something because it's really great. So this has been Rearing Rambles, and what I will say is you can look forward in the future to episodes where we cover things that aren't necessarily just a species. We will cover species still, but we will also talk about more broad topics because there aren't every species has something kind of unique about it, when you think about it long enough. But generally speaking, there's going to be things that we have to address that can apply to almost anything you're working with. And so that's why it's important to occasionally have an episode where, yeah, we're not talking about a specific species. We'll get back to that at some point because it is fun. And, we always want to have sort of a consideration for, everything around us and some of the, wonderful biodiversity we have. But we also sometimes want to get into approach. And so you're going to see more Viewing Rambles episodes where we really delve into approach. And this is one of them. So I hope this has given you something at least to think about, maybe giving you some information. And with that, I will say goodnight to everybody.